The periodic table of foods, or PTFI, Glycomics method, is a first-of-its-kind method that allows us to not only understand what types of carbohydrates at a macro level, such as starch, sugars, and fibers, are found in different food products, but actually to be able to describe them on the molecular level.
It's trying to understand what's actually in our foods, and by knowing that, that can help us further develop methods or understanding of metabolic diseases and also worldwide health.
Carbohydrates are the most abundant biomolecule in nature and the primary component of most plant-based foods. Carbohydrates are generally divided into three subtypes, including starch, sugar, and dietary fiber. Sugars consist of mono and disaccharides that make up what we generally respond to as the sweet flavors in food.
Starches are long chains of several hundred to tens of thousands of molecules bonded together in linear or branch structures. Like sugars, starch is metabolized by humans and is a major energy source.
The final group of carbohydrates found in our diet are dietary fibers. Dietary fibers are large chains of sugars just like starch. However, they contain different monosaccharides, linkages, and branching patterns that make them indigestible to humans, but allows us to nurture and feed our microbiome through those specific structures. Due to their large size, diversity in branching patterns, molecular structure, and glycosidic linkages, dietary fibers really have not been characterized on a large level.
The PTFI Glycomics method offers an integrated approach to carbohydrate analysis, where the carbohydrates in just 100 milligrams of a food sample are physically fractionated, with aliquots being taken at different steps and probed more deeply for their molecular components.
Samples first undergo preparation and processing before being hydrated in a buffer. Once hydrated, aliquot is taken for sugar analysis, which undergoes dilution, filtration before being loaded into an autosampler for analysis by ultra-high performance liquid chromatography paired with tandem mass spectrometry.
6 sugars are chromatographically separated and analyzed in just 16 minutes.
For quantitation, we compare the response of each sugar with an external standard curve that's calibrated to an internal standard. Next, for starch analysis, the fibers and starches are separated via an enzymatic digestion. Upon completion of the digestion, ethanol precipitation is used to separate the fiber fraction from the solubilized starch fraction.
The ethanol-soluble fraction containing the digested starch is dried and labeled with a reagent that enables glucose to be analyzed on a spectrophotometer. The glucose-derived starch absorbance is measured and compared to a standard for quantitation.
The ethanol precipitated fraction is then solubilized and homogenized before being separated for 2 different workflows. The fiber composition sample undergoes enzymatic digestion. The fiber linkage sample undergoes permethylation. Subsequently, both samples undergo acid hydrolysis to depolymerize the fibers into monosaccharides, which are then labeled. The fiber composition analysis is able to separate and quantify 18 different sugars.
The fiber linkage method analyzes the 30 most common linkages found in foods. Due to limited commercial standards, we normalize the fiber linkage peak areas to the fiber composition samples.
After analysis, the data is integrated, which allows us to understand how much sugar, fiber, and starch is found in each food product, enabling us to map those back to a nutrition profile. This new understanding of carbohydrates will enable scientists to understand how the precise molecular structures of different components that make up carbohydrates are able to influence not only our diets, but our microbiota composition, and also how this impacts global health.